Welcome back. So now we're going to talk about actual evolution and how it occurs. So we know what needs to occur for evolution not to work. So now let's look at the 5 agents of evolutionary change. The first is mutations. A mutation is a change at the genetic level. Mutations tend to be very random. The only time, if we are causing the mutation purposely, which we do in biotech oftentimes, dealing with other organisms, then it wouldn't be then it would not be a random mutation. But the mutations we're talking about are the ones that occur randomly. Genetic drift. So this is a change in allele frequencies due to different environmental activities. So you might have a natural disaster that kills off a large group of the population, or you might have hunters kill off a large group of a population. And that decreases the number of alleles, which then can cause problems. The founder effect is another type of genetic drift where you have a change in the gene pool because a small group from a larger population breaks off, making their own population. So the bottleneck is a type of genetic drift that is due to, typically we think of like some type of a disaster. And so one example of this is the cheetahs. They were hunted so vastly or so much that they killed off the population. So now all cheetahs are almost identical. They are so genetically similar that even producing offspring is hard because it's like, well, basically inbreeding. And so here you can see genetic drift. This is one way to look at it, where you have a large group of animals, and in the large group, only 10% of the population might have some unique mutation. If some natural disaster kills off 50% of the population, now that unique mutation becomes 20% of the population. Here's another example of that genetic drift and this is known as the bottleneck effect. And so here is an example of that. So we have a population that has a lot of similar alleles, but there are some unique alleles within that population. In this case, the yellow marbles. When something happens, only a few of the organisms escape, and in the end you end up having a totally different type of population where white and blue were the most were about equal over here. Now blue becomes the common gene and white becomes just as unusual as the yellow over here, and yellow has been wiped out. Whatever that was is wiped out. The founder effect, again, when you have a few individuals leave a population, if even one of those individuals has a unique trait, so let's look over here. Here we have a bunch of individuals and only a few leave. So we now have this one individual that maybe has a unique trait. That one individual is one in 6 individuals that have left. That trait might become very common. This is known as the founder's effect. And the disorder actually produces, and the ones that we typically look at is in a group of Amish that left a larger group, so formed their own little colony. And in that group of Amish individuals, we have a lot of people who were born with the disorder of dwarfism. So this type of dwarfism is known as achondroplastic dwarfism. Their torsos are the same size. Heads are the same size, but their limbs are much smaller than normal limbs. This also causes six-fingeredness to become more common. And so here are the 5 fingers plus this one extra finger there. That's that 6th finger. This is actually a dominant allele that we see not very often because this is, though it's dominant, it's associated with this dwarfism as well. Gene flow is the movement of alleles between populations, so individuals coming into a population while others might leave. That does occur. And then non-random mating, we choose the organisms that we want to reproduce with. We don't just randomly mate. It's typically the female is the choosy one, and the female is choosy because they have to put more energy into raising the offspring. So the best example, I'll just use humans because we all know humans. In humans, females produce a finite number of eggs, and those eggs are released each month. Depending, we tend to release only 1 egg a month. The rest of the eggs that are activated help the one individual egg that is released. And these eggs will run out, which is when we hit menopause. The egg itself is very large. It has all the organelles, has everything needed for the new organism to survive, except for 1/2 of the genes. Males produce billions of sperm daily. In 1 teaspoon of semen, you can have millions of sperm. And these sperm then move through the vaginal canal, through the uterus, to the fallopian tubes, where they will then try to get to the egg and burrow through and release their genes. Males start producing sperm at puberty and they never stop, whereas females, we produce all of our eggs before birth even occurs, and then we lose almost all of those eggs before puberty. So the number of eggs that we actually have by puberty is a very small amount compared to how many we initially started with. Females then have to house that growing embryo, fetus, until birth. So it takes a lot of energy again. Females tend to be the ones that are the caretakers. That's not always true, but many times it is. The female is the one that births the child and then cares for that child. So females tend to be the choosier of the individuals. And we choose our mates. And males also of the human species will choose. So males will look for a healthy, strong woman that can birth many children, whereas females will look for a strong, healthy man that can take care of her and her children and will give off good offspring. The last thing then is natural selection. So we have to have selection for evolution to occur, which means the environment is going to select for specific types of organisms. The best adapted individuals are going to be the ones that are going to survive and be able to reproduce, and that is natural selection. This process was explained by Charles Darwin in the 1800s. There are 2 requirements for natural selection to occur. You have to have different individuals or different types of individuals. If everyone is the same, then the environment can't select for specific individuals. and you have to have more alleles than the environment can support. So when there's a lot of organisms, and a lot of organisms of the same species, then the environment will choose those that are best suited, and the other ones will die out. I always like to talk about giraffes. So there are different, when you look at a giraffe, all giraffes have long necks, but giraffes didn't all start out with long necks. There were different types of giraffes. Some might have had short necks while others had intermediate necks. But since we had different types, variations, those that were best suited and were able to eat food off of large trees that were really, really high up, as well as off of the ground, which large necked giraffes can do, were better fit to the environment and were able to survive while the others died out. So fitness is the number of offspring that are produced. Fitness is not just being able to reproduce, but it's also being able to produce offspring that can reproduce. So my fitness is going to be determined by the number of offspring, not only that I had, but that my children have as well. The more offspring they have, the fitter I am. My genes are being passed on to more and more generations. So fitness can arise from mutations. So you have variations within fitness or within genes that can increase or decrease fitness would be from mutations that occur, from crossing over and independent assortment, which are two of the important functions that occur during meiosis to produce unique organisms or unique offspring. There are three types of natural selection that we tend to look at. Stabilizing selection occurs when, and so I'm going to just move to the next slides where you can look at this. Stabilizing selection occurs when an intermediate phenotype is favored over the extremes. So if, in this case, we're looking at clutch size for eggs, but we could also be looking at infant size in humans. So for the clutch size for eggs, if you have less than 4 eggs over here, then you're probably, because some of those birds aren't going to make it, some of the birds that do make it might end up being killed off by a predator. You're not going to be as fit. But if you have more than 5 eggs, again, some of the eggs might not make it, but you have so many that it doesn't matter. But you're not able to give all of those birds food. You can't feed all the birds that are there, and so you're more likely to have unfit offspring. But when you have just that 4 to 5 eggs here, then you're able to protect yourself from any eggs that were stolen, because now you still have a few left. And those that are here, you can make sure that they are going to survive because you can feed them properly. This can also be done looking at body size or head size. So for infants, babies that are born less than 6 pounds are typically not as healthy as babies that are born between 6 and 8 pounds. Babies that are above 9 or 10 pounds are going to potentially cause problems to mom. So really large babies can cause problems because mom may not be able to give birth. Today we have C-sections, but back in the 1800s, a C-section was very dangerous. Babies that were super small tended to be less healthy. Think of premature babies. and so they were more prone to dying also. Whereas those babies that are between 6 and 8 pounds were that perfect size. They were small enough to fit through the birth canal easily, and they were big enough to be able to survive. Directional selection then is movement of alleles directional selection is movement of alleles from one phenotype to a totally different phenotype. So you might go from small birds to large birds. That would be an example. And that could be because the environment has changed. So the example here we look at is the horse. So the initial body size of a horse was very small. They were able to move through the woods very easily, and they lived in a woody area. But as the environment changed, the size of the horse changed, and actually the number of toes that the horse had changed. So that they only now have, I believe, 2 toes, whereas they started off having, I believe, more than 2 toes. And these changes occurred because the environment changed. When you had a woody environment, you had to be small so you could fit through areas. But now that now that most horses have large fields, they don't have to be so small, and so we have changed in size. Oh, I keep doing that, sorry. All right, the last is then disruptive selection, where we are looking at either of the extreme phenotypes, and the heterozygous individual or that intermediate phenotype is not desired. This often occurs when you have an environment that has 2 very unique characteristics. So think of right by a lake. You have an open area where animals might come in, and then right behind that lake you might have a woody area. And so an intermediate phenotype might not fit in very well because they would get picked off by the individuals from the other environment. But if you have certain animals that live in the woods and another of the same type of animal that lives on the, say, sandy shore, they can survive in those 2 environments very close to each other because they can blend in. And so what we typically look at are snails. So we have snails that can blend in with, in this case, a grassy environment, and snails that can blend in with a sandy environment. But an intermediate would not be very effective because they would be picked off in both environments. Here's another example. We have lots of different types of garden snakes. Some of them are going to blend in with the grass, some blend in with dirt, some are going to blend in a sandy environment. So it just depends on where they're going to be located, what type of banding pattern they're going to have. So this is just a figure showing different types of selections for different organisms. We only have a few slides left, so I'm going to finish this up. I realize that we're at 19 minutes, but it'll only take me a few minutes to finish everything up. So we're going to look at how we maintain unique characteristics. So populations that have very little variation are not able to adapt as effectively typically. So we need to have the variation within a population, even if some of those alleles may be considered potentially harmful in certain circumstances. They may be considered beneficial in other circumstances. So mutations, of course, are going to promote variation, recombination, through crossing over and independent assortment, gene flow. All of those are going to promote variation, but some of these unique characteristics that could potentially be considered dangerous or harmful that we wouldn't want in the environment can be potentially protective in other cases. So my favorite example, the one that I know the most about, is sickle cell disease. So again, we talked about this a few times now, but in this case, sickle cell trait or the sickle cell allele is a single mutation that occurs in the hemoglobin gene, where there's just one allele has changed, or one nucleotide, I should say, has changed, and the entire hemoglobin protein misfolds. So this causes a disease that can kill you. So it's really dangerous. And in the areas where sickle cell disease is most common, there's not as many hospitals always, so people tend to be very sickly and they can die very easily. But these are the same areas where malaria is more prominent. And individuals that have sickle cell disease can't get malaria. They are protected from malaria. And because of this, this allele becomes more common. So those who die from sickle cell disease can't pass on their genes. But if you have one allele for the sickle cell disease and one normal allele, you can't get malaria and you're still healthy enough to not die from the disease. So you have the best of both worlds. You can't get sick with malaria and you can't get sickle cell disease, or you're not going to die from sickle cell disease. You have what we call the heterozygote advantage. So now let's look at what a species really is. A species is a group of subpopulations that are capable of interbreeding, and they have isolated reproductively from other species. So humans are a species, and we have and we can reproduce within the human population, but humans can't reproduce with other species, be it animals or plants or birds. We can't do that because we are isolated from being able to reproduce from them. So there are both prezygotic and postzygotic isolative barriers. And here are the different barriers. So a prezygotic barrier is something that's going to keep us from being able to have intercourse or to actually reproduce in the first place. So this might be due to the habitat. A habitat isolation means that we don't occupy the same habitat, so we're not going to come in contact with each other. Temporal isolation means that we don't reproduce at the same time. So there's a lot of organisms that have specific seasons for reproduction or different times of day that they reproduce, and so they're not going to come in contact with another species that they can reproduce with because they don't reproduce at the same time. Behavioral isolation. In animals specifically, there are courtship behaviors that occur, be it songs that birds use or gifts that insects produce to give to the female. There might be pheromones being produced. There might be fighting between animals, and the strongest is the one that the female will reproduce with. And then mechanical isolation, where the genitals won't go together. And so genitalia, that's interesting because there are some genitals that are so unique. I believe it's the duck that has a spiral-shaped penis, and the female has a spiral-shaped vagina. So they go together perfectly, but no other organism could reproduce with them. Postzygotic isolation, the gametes can't reach each other. So sperm can't find the egg, or the sperm can't get to the egg. the zygote that is produced dies. So as soon as fertilization occurs, the zygote doesn't have enough genes, or something happens, but the zygote can't survive. Hybrid sterility, so the zygote survives and offspring are produced, but the offspring are infertile. Or the hybrid is fertile but can't produce very viable offspring. So their offspring are not as fit, and they themselves can't produce offspring very effectively. This again. So how do we actually create new species? This can occur over a long period of time, or this typically occurs over a long period of time. When we think about speciation, we're not looking at like within a day or two. When two or more species branch from a parent species, that's known as speciation. When reproductive isolation develops, that's a speciation. And there are two basic mechanisms that happen to cause speciation. Either allopatric speciation, where a barrier is formed, so the continental drift produced a very large barrier within populations because of the ocean. The populations that may have been the same species at one time may, because of the isolation, may no longer be able to interbreed. And then sympatric, some genetic change occurs, a mutation occurs within some organisms in a population, and that mutation causes a reproductive barrier to be put in place. So here we have both allopatric and sympatric speciation associated with trees. And so here's the parent trees, and then over the hundreds of thousands of years, mountains form, and the trees are separated and no longer can, when they're brought back together, can breed with each other. Here we have sympatric speciation, where a population of trees, so typically you start with one tree that has mutated and no longer can reproduce with others, and so instead fertilizes its own cells, producing new offspring from itself, forming a new species. And here you can see allopatric speciation in foxes. So we have the Arctic and the gray fox. Early on, they were both within the same fox population, but as the time went on and the foxes spread to either the northern or the southern regions, they became different uniquely from each other. So the gray fox adapted to heat, while the Arctic fox adapted to the cold, and they no longer interbreed with each other. This is an amazing video. It's about 9 minutes, but it's a really great video. The video is called SciShow, and I would highly recommend you watch this. I'm not going to watch it on here like I've done in the past. I'm just going to have you watch this on your own. But that's all I have for you. And so I'm going to stop here, and I will post this video. Have a wonderful day. Bye.